biologists, welcome to today's daily A-Level Biology revision task. In today's task, you are asked to produce a concept map on the roles of mammalian sensory receptors in converting different types of stimuli to nerve impulses. The following short video will go and outline the role of the Pacian corpuscle in producing sensory impulses and generator potentials.
Right, so your placental corpuscle, you've got the end of the neuron, right, and the corpuscle, right, so I've only done a small bit of it, but your corpuscle would kind of go and surround it, right? So it goes and it surrounds the neuron membrane. You've got the pressure that goes and it deforms it, so the pressure is acting as a stimulus. When the pressure acts as a stimulus, that stretching physically stretches the neuron membrane. It stretches it open, right? So it causes proton channels, right, to become wide enough to let sodium ions in. Because usually they're too close together, so the gap in them is too close to let sodium ions come in. So the first part that kind of happens is that in a normal state, the stretch-mediated sodium channels in the sensory neuron membrane are too narrow to allow sodium ions through. Right? So the neuron in the central corpuscle has a resting potential. Right, that resting potential, right, because if you think about it, we always talk about nerve impulses being electrical like signals. They kind of are, but it's all to do with the potential difference between the inside of the neuron and the outside of the neuron. Right, so on Friday we'll look at resting potentials and stuff. But it's all to do with the amount of positive ions inside in comparison to the amount of positive ions outside. Right, so that's all resting potential is. And it's about minus 70 millivolts is the resting potential of it. But we'll talk about that next lesson. So the second thing that happens, so when pressure is applied, the placental corpuscle changes shape, right? So this causes the membrane around the end of the neuron to be stretched, right? If we go in and we're stretching that membrane, right? So when the membrane stretches, the sodium channels are pulled open wider, right? So if the sodium channels are pulled open wider, sodium can now move into this neuron, right? They're going to be moving in by diffusion, they move down concentration gradient. So it's not taking any energy, it's not by active transport. Because it's straight diffusion, and because it's moving down the concentration gradient, that's why you're getting a really fast influx, right? So the influx of positive sodium ions changes the potential difference of the membrane, so it becomes depolarized. Right, all depolarized means is that the charge is pretty similar on both sides. Polar means different, dipolar means similar. Right, so this results in a generator potential being produced. And it's the generator potential that goes on to go and it forms the action potential that moves all the way down your neurons, all the way back to your brain eventually. Right, we'll look at action potentials next week. So, in turn, the generator potential causes the action potential, right, we just talked about that, and eventually that action potential is transmitted all the way along the neuron. Right, just to give you a little bit more information, after your pressure's been applied, right, when pressure's stopped being applied, it goes in this placental corpuscle, bounces back into its normal like shape. When that happens, it stops stretching this membrane of the neuron, so all of these sodium channels all shut. So sodium can no longer move back into or move into the neuron. The sodium will then be actively transported out, which goes and it resets this resting potential, so it repolarizes it. Another thing is, you know, if you carry on putting pressure down on something, so you know if you go and you sleep on your hand and you wake up, you can't feel it, it feels dead. That's because your sodium channels in like your neurons in your finger, so in your pressure receptors, have just been left open too long, so there's no longer any potential, right? So you can no longer form and generate potential in your nerves, in your finger or your hand or whatever you sleep on, which is why it feels dead. Right.
Hopefully this video should have helped you complete your daily revision task. Please subscribe so you can keep up to date with other videos to help with your daily revision tasks. You can follow me on Facebook or follow me on Twitter to help you keep up to date.